Take two patients standing side by side. One is outwardly heavy, but maintains stable blood pressure and normal glucose levels. The other appears completely lean, yet requires medication for hypertension and shows clear signs of prediabetes. Medical science identifies this second condition as the TOFI phenotype, thin outside, fat inside. In these cases, an outwardly healthy aesthetic masks severe internal pathology. If we bypass the skin and inert subcutaneous fat, we find a throbbing, vascularized, inflamed mass of visceral adipose tissue, tightly packed around vital organs. This mass functions as a hyperactive endocrine organ, continuously secreting over 600 distinct proteins and inflammatory signals directly into the body. The number on a bathroom scale is merely a cosmetic illusion. The hidden volume of your visceral fat acts as the true biological driver of cardiometabolic disease. Visceral fat evolved as a specialized adipoimmune organ. The omentum, the large fold of fat draped over the stomach, originally functioned as an ancient filtration barrier designed to neutralize bacteria leaking from the gut during periods of famine. Modern diets, dominated by ultra-processed foods, degrade the intestinal wall. This creates a leaky gut, subjecting the abdominal immune system to a persistent, unnatural influx of bacterial fragments. Molecules known as lipopolysaccharides breach these degraded tight junctions. This exposure triggers metabolic endotoxemia, trapping visceral fat in a permanent state of low-grade inflammation. Aggressive M1 macrophages swarm the visceral fat depot in response to the breach. They convert the tissue into a toxic factory, churning out inflammatory cytokines like TNF-alpha and IL-6. Modern diets have hijacked this ancient survival mechanism. Our specialized abdominal immune system now functions as a self-destructive engine. Unlike subcutaneous fat, visceral fat routes its toxic secretions directly into the portal vein, a high-volume pipeline straight into the liver. This floods the liver with excess-free fatty acids, slowly engorging healthy tissue with yellow lipid droplets. This continuous exposure results in hepatic lipotoxicity. The liver becomes insulin-resistant, driving the development of non-alcoholic fatty liver disease. When safe subcutaneous depots reach their storage limit, surplus toxic lipids spills over into the pancreas. This toxic accumulation induces severe local oxidative stress that actively destroys the pancreatic beta cells, responsible for regulating blood sugar. Visceral fat's danger is rooted in its privileged anatomy. It poisons the liver and the pancreas long before the rest of the body can mount a defense. Chronic psychological stress and sleep deprivation hyperactivate the brain's HPA axis. This results in a sustained, systemic release of the stress hormone cortisol. Visceral fat utilizes an enzyme called 11-beta-HSD1 to amplify its own growth. The enzyme grabs inactive molecules from the bloodstream and reanimates them into active cortisol right inside the fat cell. This creates a localized Cushing state, where cortisol concentrations inside the visceral depot are massively amplified compared to the rest of the body. National health data proves that sleeping fewer than 6 hours per night drastically accelerates this enzymatic fat accumulation loop. Visceral fat functions as a rogue stress organ. It uses 11-beta-HSD1 to self-amplify its own growth whenever the host is sleep-deprived. The toxic secretome of visceral fat eventually escapes the liver. It enters the systemic blood circulation and initiates the foundational steps of cardiovascular disease within the arterial walls. The expanded fat depot overproduces a protein called PAI-1, which inhibits the body's ability to break down clots. This creates a highly prothrombotic environment that correlates strongly with coronary artery disease. Identifying this hidden threat requires clinical diagnostic tools that can see past traditional metrics like BMI or waist circumference. The gold standard for quantifying visceral fat area in TOFI patients is a cross-sectional CT scan targeted at the L4-L5 lumbar level. Imaging the internal organs directly reveals the inflammatory phenotype of the intra-abdominal cavity, exposing the true atherosclerotic threat hidden from standard clinical metrics. Visceral fat is highly metabolically active. This property makes it exceptionally responsive to targeted biological interventions. Research shows that high-intensity interval training is significantly more effective at mobilizing intra-abdominal fat than moderate steady-state cardio exercise. The near-maximal effort of HIIT produces massive surges of catecholamines like epinephrine. These molecules bind to specialized receptors on the visceral fat cell membrane, triggering rapid lipolysis and causing the cells to deflate. Sustained reduction requires a diet rich in fiber and polyphenols. These nutrients repair the gut microbiome and shut down the endotoxemia that fuels visceral inflammation. Optimizing human longevity requires moving beyond weight loss targets to focus on biological strategies that shrink and disarm this invisible endocrine organ.